Look around your city for a second.
Yeah, just like take a really close look.
Exactly. Like whether you're commuting or, just looking out an office window or walking down the street, if you really look at the buildings, what do you actually see?
You're mostly seeing concrete, steel, and a lot of glass.
Right. You see static, unchanging, essentially dead materials.
Hmm.
And the wild thing is those dead materials, just by existing and, well, by being manufactured in the first place, they're responsible for nearly 40% of all global energy-related CO2 emissions.
It's massive.
It is. I mean, the very act of building our world is, it's suffocating it. And this is why we're here today. What if the concrete and glass around you weren't dead? What if they were like alive, breathing, and actively reversing climate change?
I mean, I know it sounds like pure science fiction, but looking at the data, it is rapidly becoming an engineering reality. We are looking at a fundamental shift in how we conceive of human habitation.
Yeah.
We're moving away from structures that just occupy space, to structures that actively participate in the environment.
Which is just mind-blowing. So, welcome, you ever-curious learner, to today's deep dive. Our mission today is to extract the ultimate aha insights from a really fascinating stack of sources we've compiled for you.
We've got a lot of ground to cover today.
We do. We've got recent architectural reviews, metabolic engineering studies, NDPI research papers, and this brilliant climate tracker report from WION. And what they all point to is the science and the future of bio-integrated architecture.
Right. So we are talking about liquid trees, CRISPR-engineered super algae, and like genuinely 3D printed living buildings.
Living buildings, it's crazy.
Yeah, we are essentially talking about transitioning our cities out of the Anthropocene era.
Which is that destructive, human-centric era, right, where we just extract and take from the earth.
Exactly, transitioning from that destructive legacy into a regenerative Ecocene era. In the Ecocene, our structures participate in the urban metabolism, they consume waste, they produce clean air, and, they grow.
Okay, let's unpack this. Because to understand how we get to massive living skyscrapers, we first have to look at the street-level technology, right? We need to look at how water and microscopic algae are, well, actually outperforming traditional urban greenery right now.
Yeah, and a perfect starting point from that WION report is the liquid tree concept, specifically the Y algae photobioreactor project that scientists developed in Argentina.
Right, the Y algae project.
If you picture a street-level installation, it looks like a sleek, illuminated, kind of futuristic tank filled with green liquid.
Like a glowing sci-fi tube on the sidewalk.
Yeah, pretty much. And that liquid is really just water teeming with native microalgae. And because it's a closed system, it completely controls the light and nutrient delivery to maximize the whole biological process.
And its job is beautifully simple, right? It breathes in carbon dioxide and breathes out oxygen. But what really blew my mind from the data is the sheer scale of its efficiency. Each one of these single standalone devices can capture about 0.5 metric ton of carbon dioxide annually.
Right, which is just a staggering number when you consider the actual footprint of the device. Now, I should clarify, the researchers are very explicit in the literature about this. This is not meant to replace real trees in nature.
Oh, totally.
If you have the soil and the space to plant a traditional oak tree, you should always plant the oak tree.
Obviously, yeah.
But think about heavily paved, human-altered urban environments. We were talking about, underground parking garages, concrete traffic medians, subway entrances.
Places where the root system of a giant tree simply cannot survive. I mean, you can't just slap a forest into Times Square or like on a subway platform.
Exactly.
So placing these liquid bioreactors in a concrete median is basically like giving the city a localized dialysis machine, right? Pulling the ambient, polluted air through a filter, scrubbing the carbon out of it, and pumping clean oxygen back onto the street level.
A dialysis machine is a highly accurate way to visualize that mechanism. And the reason this dialysis is so effective is that, well, a microalgae's chemical efficiency for photosynthesis is incredibly potent.
Why is that?
Because they are single-celled organisms suspended in water, right? So they don't have to waste energy growing heavy trunks or deep roots or complex branch structures like a regular tree does.
Oh, that makes sense. They don't need the physical architecture.
Right. Almost every ounce of their energy goes directly into photosynthesis. This makes them roughly 10 to 50 times more efficient at capturing carbon than terrestrial trees.
10 to 50 times.
Wow.
Wow. And architects aren't just leaving these biological dialysis machines on the sidewalk either. They are scaling this up to the buildings themselves through bio-integrated facades. We're seeing a leap from a standalone tank to the actual building envelope.
Yeah, take the BIQ house in Hamburg, Germany, which is prominently featured in our architectural reviews stack. It was built with flat photobioreactors integrated directly into the facade.
So the whole building is covered in them.
Basically, yeah, the building is essentially enclosed by these transparent glass panels filled with microalgae. But it's not just sitting there looking green, the system is dynamic. The panels can actually rotate to track the sun, maximizing solar exposure.
That's so cool.
And as the sun hits the panels, the algae multiply rapidly.
And that rapid growth provides multiple passive benefits, right? Like the thick green algae layer actually acts as dynamic shading for the building, keeping it cool in the summer.
Yeah, it replaces traditional blinds or tinted glass.
Right. Then, the solar energy stored in the panels, plus the grown algal biomass itself, supplies renewable energy for the building's operation. I read that they actually harvest the algae, scrape it out of the panels, and send it down to a bioreactor in the basement to generate heat. Is that right?
That's exactly right. It provides climate control, it generates its own combustible biomass for heating, and it sequesters carbon all at the exact same time. It's a closed-loop ecosystem functioning as a human residence.
It's incredible. And there's another amazing example from the sources, too. A student residence in Paris that's slated to feature a variation of this technology.
Oh, yes, the Paris project.
They are using standard double glazing for the windows, but with an extra layer filled with warm water and microalgae connected to a pump system. It heavily insulates the building, which could potentially cut energy consumption by 50%, all while the algae sequester carbon right there in the window you are looking out of.
And if you look at the numbers, depending on the operational modes and the growing environments, these algae-integrated buildings can sequester an average of 5 grams of CO2 per square foot per day.
Wow.
Multiply that by the massive surface area of a modern skyscraper, and you have a massive carbon sink in the middle of a city.
I have to ask the obvious question here, though. Why? Like, why are these microscopic green blobs so much better at capturing carbon than a massive plant? I mean, a tree is huge. It has millions of leaves specifically evolved to catch the sun and air. How does a single-celled organism beat it so soundly?
Well, answering that requires zooming in past the water, past the cell wall, down to the molecular biological engine of the algae itself. The secret weapon here is a specialized cellular organelle called the pyrenoid.
The pyrenoid. Okay, that sounds like a sci-fi villain's doomsday device.
It does sound a bit intimidating, but it's really the hero of our carbon capture story here. To understand why the pyrenoid is so vital, we have to look at the enzyme in all plants and algae that's responsible for carbon fixation. That enzyme is called rubisco.
Rubisco.
Yeah. Rubisco's job is to grab CO2 out of the air and stitch it into organic sugar. But the problem is, rubisco has a major evolutionary flaw.
Yeah, the studies mentioned it has a very low affinity for CO2, which honestly seems really counterintuitive for an organism whose entire job is to eat CO2.
It does, but it traces back to when rubisco evolved billions of years ago. The ancient Earth's atmosphere was incredibly rich in CO2 and had very little oxygen. So rubisco never had to be particularly precise. It could just blindly grab at the air and almost always catch a carbon molecule.
Oh, because carbon was everywhere.
Exactly. But today, our atmosphere is oxygen-rich. So rubisco is clumsy now. It often grabs an oxygen molecule by mistake, which forces the plant to undergo a wasteful energy-draining process just to spit the oxygen back out so it can try again.
So rubisco is essentially a poorly designed claw machine at an arcade. Like, it keeps dropping the prize because the claw is too loose, and half the time it accidentally pulls up the wrong stuffed animal anyway.
That is a perfect analogy for the mechanical failure of rubisco. It really is like a bad claw machine. And to fix this catastrophic inefficiency, algae evolved the pyrenoid.
Okay, so what does the pyrenoid do?
The pyrenoid acts as a biological pressure chamber. It creates a localized, highly concentrated CO2 microenvironment directly surrounding the rubisco enzyme.
Whoa.
Yeah. By sealing the rubisco inside this high-pressure carbon bubble, it essentially force-feeds the CO2 to the enzyme. Your claw machine can't miss because it's completely buried in the exact prize it needs.
That is brilliant. Nature engineered a structural workaround for a flawed enzyme. And if you're the listener walking past one of these biofacades, you aren't just looking at green water. You are looking at trillions of microscopic pressure chambers working simultaneously, force-feeding themselves the exhaust from the cars driving by you.
Exactly. That carbon concentrating mechanism is exactly why algae can fix carbon so much faster than land plants, which, for the most part, completely lack this specialized pressure chamber.
But Nature didn't engineer algae to live inside the walls of a skyscraper in a modern megacity. Urban environments are incredibly harsh.
Very harsh.
You've got extreme summer heat waves radiating off the asphalt, severe air pollutants from diesel engines, and freezing temperature drops in the winter. We can't just scoop some wild algae out of a pond somewhere, slap it in a glass panel, and expect it to survive, right?
No, absolutely not. Wild strains would experience immediate culture crashes. The temperature swings alone would either boil or freeze the cells. And this is where the research pivots heavily from natural biology to synthetic biology and advanced genomic engineering. Scientists are now using CRISPR-Cas9 genome editing tools to create designer superalgae.
Superalgae. And from reading the metabolic engineering studies in our stack, these CRISPR upgrades are highly specific. They aren't just trying to make the algae generally tougher or whatever. They are going into the genetic code and tweaking specific metabolic pathways.
Yes.
For instance, they are modifying these strains to have extreme temperature tolerance, so a 100-degree summer heat wave hitting a glass building facade won't just cook the culture alive.
Right, they have to engineer that resilience. They're also enhancing lipid and biomass production at the genetic level, forcing the algae to generate more usable material faster than it naturally would.
Which makes the building more efficient.
Exactly. And they're even engineering strains that can thrive directly on power plant flue gas. That means the algae are genetically programmed to eat highly concentrated toxic industrial emissions, which would kill wild algae instantly, by the way, and convert those toxins into harmless biomass.
Okay, but wait. If we are pumping billions of genetically modified superalgae into the walls of residential buildings, we have to talk about containment. What's stopping a mutant algae strain from escaping or mutating further? I mean, having genetically modified sludge leaking out of a broken window and into the city water supply feels like the inciting incident of a disaster movie.
It does sound scary, and this raises an important question. The biosafety protocols detailed in the research are incredibly rigorous to prevent exactly that scenario. First off, these are closed-loop photobioreactor systems, which provide strict physical containment. The water isn't exposed to the open air.
Right, it's sealed.
But the biological containment, how they manipulate the genetics to ensure safety, is far more fascinating.
Yeah, from the MDPI research papers, they achieved this biological containment by utilizing genomic safe harbor sites. So when scientists use CRISPR to edit the algae's DNA, they don't just randomly insert the new genetic instructions and hope for the best.
No, not at all. They insert the new traits into specifically identified safe harbor sites within the genome. Think of these sites as secure, isolated vaults within the DNA.
Okay, vaults.
Yeah. When a transgene is placed in a safe harbor site, it is highly stable and expressed safely without disrupting the organism's core functions. And most importantly, traits placed in these vaults are completely immune to horizontal gene transfer.
Meaning, even if there was a physical leak, say a panel breaks, and the superalgae mixed with wild bacteria in the city's water supply, the engineered traits, like that extreme heat tolerance, cannot jump from the algae to another organism.
Precisely.
The genetic code is locked in a vault that can be read by the algae, but never copied or shared with anything else.
That's a great way to put it. Furthermore, the systems are monitored by specialized detection assays. Rather than throwing dense acronyms around, you can think of this technology as a microscopic biological spell checker.
A spell checker.
Yeah. Specifically, isothermal RPA CRISPR-Cas monitoring systems. It continuously scans the algae's genetic code in real time. If it detects that the DNA has mutated away from the safe, programmed baseline, it triggers an alert or a fail-safe to neutralize the culture.
Oh, wow.
So the system ensures the living skin of the building remains healthy and entirely contained to its original programming.
Okay, so we've managed to keep these hyper-efficient, highly regulated superalgae alive and contained in liquid tanks and window panes. But water and glass panels don't hold up roofs. How does this soft, watery matrix become actual load-bearing architecture? Like, how do we actually build a physical structure using a liquid organism?
This is where the realms of biotechnology and robotic additive manufacturing, which most people know better as 3D printing, collide. To move beyond just decorative or insulating glass panels, builders and researchers are now 3D printing with algae-laden hydrogels.
Hydrogels. We see hydrogels used all the time in medical applications, right? Like
Yeah.
the material that makes up soft contact lenses. It's flexible, holds moisture, but it still maintains a structure.
Exactly. The building applications use a similar concept, specifically alginate-based hydrogels like Pluronic F127. They use biologically inert materials that won't harm the algae. So instead of pouring a foundation with a cement mixer, massive robotic arms extrude this gel layer by layer. The hydrogel is mostly water, about 85 to 90%, but it can be printed into complex, three-dimensional open lattice structures.
And since the gel is mostly water and it's completely transparent, I imagine that solves the light and nutrient problem, right? If they print it as an open lattice, like a rigid sponge, the algae deep inside the center of the structure can still photosynthesize.
You hit the nail on the head. The geometry of the lattice is specifically inspired by aquatic structures, like sea sponges, to allow passive capillary transport of nutrients.
Passive transport, so no pumps.
Right, no mechanical pumps. It acts exactly like a sponge, drawing water and food up through the microscopic channels of the structure naturally. Studies have shown that algae encapsulated deep within these 3D bioprinted structures can survive, grow, and photosynthesize for over a year.
That is an incredible feat of biology, for sure. But a hydrogel, no matter how clever the sponge-like lattice is, isn't going to hold up a skyscraper floor. You can't build a load-bearing wall out of something with the consistency of a contact lens.
Well, no, you can't. The gel itself is really just the temporary scaffolding. The ultimate breakthrough here, the chemical mechanism that turns this soft gel into actual structural architecture, is called MICP.
MICP.
Yeah, that stands for microbially induced calcium carbonate precipitation.
Here's where it gets really interesting, you guys, because MICP relies on the fact that algae feature a dual carbon storage mechanism.
Right.
We already spent time talking about the organic pathway, which is photosynthesis, where the algae use their pyrenoid pressure chambers to absorb CO2 and turn it into soft organic biomass. But algae also have a secondary inorganic pathway, right?
Yes, they do. Through their regular microbial metabolism, the algae naturally elevate the pH level within the watery hydrogel. And as the pH rises, the concentration of bicarbonate ions in the gel increases. The researchers then introduce calcium ions into this high pH, high carbon environment.
And when those calcium ions hit the carbon in that specific pH, a chemical reaction occurs. The result is the precipitation of calcium carbonate. For anyone who isn't a geologist, calcium carbonate is essentially limestone.
Exactly. The algae mineralize the CO2 they've captured from the air into a stable, solid carbonate rock. This mineral phase mechanically strengthens the soft bioprinted hydrogel structures. So over time, the soft sponge turns into stone.
So they aren't just sitting in a gel looking pretty. Through this MICP process, the algae are actively 3D printing their own skeletons.
Yeah.
It's like the building is a living coral reef. A coral polyp is a tiny, soft organism, but it pulls minerals from the ocean to build a massive, rock-hard reef around itself. These biobuildings are doing the exact same thing, literally pulling carbon pollution out of the air to build their own structural reinforcement.
And if we connect this to the bigger picture, we transition architecture from an extractive process to a restorative one. For centuries, we have mined limestone out of the earth, burned it at massive temperatures to create cement, and released billions of tons of carbon into the atmosphere in the process.
It's just a cycle of destruction.
Right. But with MICP, we start mining the earth for dead materials. We start partnering with biological processes to grow living infrastructure that permanently locks carbon away in the form of building blocks.
Okay, so what does this all mean for you, the listener? If you're listening to this, maybe you live in an apartment building or you commute to an office tower. The buildings of the future won't just be static boxes where you sit at a desk or sleep. They will be active biotechnological engines.
They will be entirely integrated into the urban ecosystem, functioning almost like an organ within the body of the city.
Right. Imagine washing your hands or taking a shower, and the wastewater doesn't just disappear into a dark municipal sewer miles away. Instead, it is routed directly through the transparent facade of your building to feed the algae lattice in the walls.
Yeah, a fully circular system.
And in return, that algae utilizes the nutrients to actively clean the smog out of the air you breathe on your balcony.
It's also an economic engine. These designer CRISPR algae produce high-value downstream products. Depending on the strain engineered for the building, the biomass can be harvested and processed into biofuels, industrial pigments, omega-3 oils, or even bioplastics.
The sources suggest that the biomass harvested from the walls of your building could literally be sold back to the grid to offset your rent or the building's maintenance costs. Your home becomes a biofactory that actually pays for its own upkeep.
Now, it is important to acknowledge the current hurdles. The research highlights that high initial capital costs for these robotic printing systems, the complex downstream processing required to turn the biomass into usable goods, and incredibly strict government regulations regarding genetically modified organisms are, well, they're significant barriers to immediate widespread adoption.
Of course. It's not happening tomorrow.
No, but the rapid fusion of these distinct fields, CRISPR gene editing, AI-driven bioprinting, and scalable photobioreactors, means that this reality is much closer than we think. Pilot projects like the BIQ house and the microalgae window panes in Paris are already proving the concept works in the real world.
It really makes you rethink the entire definition of human progress. For thousands of years, our progress has been defined by taking things out of the earth, carving stone, smelting iron, pumping petroleum, and turning them into dead monuments to our own engineering prowess.
That's been the whole story of architecture.
Right. But as we step out of the Anthropocene and into this new Ecocene era, the ultimate status symbol of tomorrow's cities won't be how tall a building is or how much imported steel it uses. The status symbol will be how alive it is.
Definitely.
Just imagine looking out your window in the morning, not at a skyline of static gray glass reflecting the sun, but at a breathing, growing biosynthetic forest that actually changes color as it absorbs the city's morning exhaust.
It's a beautiful vision.
It really is. And it leaves you with a profound question to mull over as you walk past the concrete and steel today. What kind of relationship will you have with a home that actually requires you to tend to it so that it can tend to you?